The overall goal of this procedure is to measure the cytotoxic activity of antigen-specific CD8 T cells using a cell-based flow cytometry assay. This is accomplished first by expanding Effector's CD8 T cells for 6 days in the presence of the peptide of interest. In the second step, target CD4 T cells are pulsed with the peptide of interest and then mixed with unpulsed CD4 T cells. Next, the effector CD8 T cells and target CD4 T cells are co-cultured for 6 hours at different effector-to-target ratios. In the final step, the killing of the peptide-loaded target CD4 T cells and the number of effector CD8 T cells are quantified by flow cytometry. Ultimately, the intrinsic killing capacity of the antigen-specific CD8 T cells expressed in lytic units can be calculated.
The main advantages of this technique over existing method like the chromium release assay is that this technique uses autologous primary target cells instead of immortalized cell lines. And the actual target cell death and effector cell numbers are quantifiable at the single cell level.
Pearline now, research assistant in my laboratory, will be demonstrating the procedure.
To isolate the CD8 positive effector cells, begin by thawing autologous cryopreserved PBMCs in a 37 degrees Celsius water bath, and then wash the cells in up to 50 milliliters of complete media. After counting, dilute the PBMCs at a concentration of 5 million cells per